Artificial life, evolution, and the origins of multicellularity. Hi, everyone. I'm Mihai Shishak, and today I'm thrilled to share my simulation project with you, a passion that has been 10 years in the making. Artificial life combines my two greatest passions, nature and computing. This field uses computer simulations to study systems that mimic natural life, its processes, and its evolution. The term was first coined in 1986 by Christopher Langton, a computer scientist. By recreating biological phenomena in silico, artificial life researchers gain new insights into traditional biology. Perhaps the most well-known life simulator is Conway's Game of Life. It uses a two-dimensional grid and a simple set of rules to simulate cellular behavior. Despite its simplicity, the system produces astonishingly complex, self-replicating structures, serving as a prime example of emergent behavior. My biggest inspiration for this project came from David Attenborough's First Life. I wanted to create a simulation that captures the transition from single-celled organisms to multicellular species, exploring how entire ecosystems evolve. While many simulations focus on either grid-based systems or fully formed creatures, I felt there was a gap. I wanted to focus specifically on the boundary between single-celled and multicellular life. How did multicellular life begin? How do cells cooperate to form an organism? How many different paths could multicellularity take? These are some of the big questions I want to explore. And because this evolutionary history isn't preserved in the fossil record, we can only theorize about the answers. There are several real-world biological theories and species that I hope my simulation will eventually be able to replicate. The colonial theory, the idea that multicellular life evolved from groups of single-celled flagellates coming together. Dictyostelium discoideum, a unique amoeba that can act as a single-celled organism, a multicellular slug, or even multicellular fungi depending on its life stage. Volvocene green algae, a lineage often studied as a model for multicellularity, beautifully illustrating a possible evolutionary path for plants. Trichoplax, a simple multicellular animal with no symmetry, composed of only 6 cell types. Simulating something this minimal feels achievable. Biofilms, communities of bacteria that band together, multiply, and spread, a kind of microbial collaboration. To simulate these systems, I model individual cells and their organelles. I don't hard-code multicellularity into the simulation. Instead, each cell decides which organelles to grow and how to function. I want both single-celled and multicellular organisms to emerge and thrive based on environmental pressures, with no predetermined outcomes. The simulation is set around deep-sea hydrothermal vents. At the start, random species are generated and introduced. Most of this DNA is junk, so many species go extinct quickly. But some survive, and from these survivors, complexity emerges. In the early stages, simple organisms dominate, feeding on gas bubbles or dead cells with specialized organelles. Mutations during cell division create variation, and over time, evolution takes hold. As resources grow scarce, species begin to differentiate and carve out unique niches. Evolution is a game of numbers. Running simulations of 200,000 cells on a GPU for weeks consistently shows similar results. Cells evolve increasingly efficient organelles, and primitive multicellularity begins to appear. At first, cells simply stick together, but over time, they evolve to share energy and work cooperatively. Extinction events and subsequent repopulation spur further adaptation, and the simulation captures the dynamic complexity of artificial life. The newest 0.7 release introduces exciting features, including a real-time tree of life visualization inspired by biology. This tree sits alongside the simulation and shows the relationships between species. The center of the tree represents the past, while the edges show the present. Each slice corresponds to a species, with tangent lines tracing their evolutionary origins. Another new feature lets you analyze save file statistics. When you run the simulation for weeks with autosave enabled, you end up with tens of gigabytes of data. The new tools make it easy to visualize key moments, like extinction events or diversification spikes, directly from the save files. In one test, I disabled the flagellum organelle to see how species would evolve without it. This run, which spanned from October 13th to November 5th, included 282 save files. The data revealed interesting trends, including spikes in cell populations driven by experimental attempts at multicellularity. Some species exploited quirks in the physics engine to form massive cell clusters that created a vacuum like effect, drawing food from across the simulation. These clusters acted almost like primitive sponges, with boundary cells consuming food and transferring energy to interior cells. However, the formations were unstable, as their sheer size overwhelmed the collision logic, causing them to collapse. While these clusters exploited the physics engine, they represent a promising step toward evolving stable multicellular life. These behaviors also stress test the simulation, helping me identify areas to refine. It's incredibly motivating to see progress toward evolving the first sponge like organism organically. I know this simulation is a niche project, but it's unmatched in its features. While it's still in early access and far from finished, I'm excited about its direction. To help you decide if it's right for you, I've released a free demo. The demo has limited world size, no DNA editing, and doesn't include the built-in save files featured in the trailer. But it's free to try. As a solo developer, I'm highly engaged with the community. I regularly monitor the Steam forums and the EvoLife subreddit for feedback. Let me know how you're using EvoLife and what features you'd like to see. I'm always listening. So head over to Steam, download the free demo, and let your GPU heat your home this winter while you explore your own digital microbial evolution aquarium. Thanks for watching, and happy experimenting.